The databases, how we use sequence ontology terms to do this. And I'm gonna, this is gonna be followed by taking you to the genome browser using a gene as an example, and show you how you can get to display all variants for a given gene or transcript, get a table with all the SNPs, visualize a structural variation, download an alignment, explore a specific variation, uploading data of your own, using a couple of tools that we have to analyze variation data, and how to get in touch with us. Over here, I have some of the kinds of variation that you're going to be introduced today. We have single nucleotide polymorphisms for 12 different species, and structural variants for Arabidopsis thaliana and sorghum bicolor. The majority of these fall under the naturally occurring variation, but we also offer mutagenesis-induced variants for sorghum. The last type of variation that we have is interhomeologous variants, where the components of the genome components of the wheat, the genome, have been aligned with each other, as well as barley and bracky, to identify a nucleotide level variants. So here is just a summary table where we have the 12 species that we have data for. This is it only includes the name a key for the study or the project that is taking that is producing this data. At the end of this webinar, I have a table with full details about number of variants, the populations that were genotyped, etc. What's most interesting is at this point we have over 230 million plant SNPs in our databases, and a couple of interesting details come from the maize variation set at pancia.org, and this because it includes over 16,000 maize accession lines. And for tomato, we have the largest number of SNPs coming to 71 million, and these have been genotyped in 84 lines by the 100 Tomato Genome Sequencing Consortium. The way we obtain this data, this is on the top part, you have this is work in progress with our collaborators of Cyverse, prior iPlant, where they are going to facilitate submission of variation data to us and to other databases as well. But currently, we have a very productive collaboration with the European Variation Archive and the Transplant Group, both at the EVI, based at the EVI, and we receive data directly from metadata, from VCF formatted files and metadata. Our databases are loaded using the two kinds of files that I mentioned, and each variant gets assigned a stable ID. You will be able to obtain individual genotypes as well as population frequencies for allele and genotype for all the data. And we use a variant effect prediction algorithm to classify SNPs based on the predicted functional effect on the transcript using sequence ontology terms. By using controlled vocabulary and standards, this supports the mining of complex data. Some example of we call this the VEP pipeline, and the SO classifications include, say, a variant that is in that maps to an intron, a variant that is causing a missense mutation, one that introduces a stop in the sequence, or one that alters the splicing sites of the predicted peptide. So this is the homepage of the Gramene website, and to go directly to the variation, you will need to click into any of these options, the genomes link on the left navigation bar, the Genome Browser at the bottom is a link to the portal, or either the icon or the name for genomes. This will take you to the species, to the genomes page, which is a mirror of our Plants Ensemble org, almost a mirror, because our databases are identical. We share, we produce the data jointly, process and produce. And they are, we use the Ensembl infrastructure. So here I'm only want to give you a flavor of the type of images and text downloads that you can get for variation. In this particular slide, you are seeing a gene that has 2 transcripts in red, and the variations, individual SNPs, are shown as ticks up here in the first track, color-coded, and you have a legend that tells you what kind of predicted functional effect will have, could have this variant, and it is interspread with protein InterPro domains. From this, for this same gene, you could decide to align the sequence against another related plant species, and have show the regions that are conserved, as well as the variants that are that have been identified, and see whether they are also conserved or not. And finally, this is just one table where you can see the population frequencies, in this case for genotype, and also alleles. Sorry. Okay, the example that I'm going to use to show you, I'm going to use screenshots to show you step by step how you can get all of the images similar to the one that I showed you before. And the gene that I chose is the CLE18, which is the clavata3 ESR-related peptide. These are involved in plant growth and development. And I chose this gene because there is a study that already, it was simple. I could mine from one of the supplementary tables that it has 2 SNPs with variant effect predictions affecting the stop codon. From this table, I cannot, it doesn't say whether it's introducing a stop or removing a stop codon, but that's what we're going to find out. So first, we're going to go to the genomes homepage, and we're going to type in the search box here or over here the name of this gene, CLE18. When we do that, we get a series of results, and we're going to choose the one for Arabidopsis thaliana. We could click either on the gene directly or go to the location of this gene. But for our purposes, I'm going to click on the gene page, and this is going to open a page, a view in among other available views for this particular gene. Over my left, I see a sequence tab, and this is the one I'm going to choose to get the sequence of this gene. Let me go back here just to show you that this gene is a small sequence, barely over 300 base pairs, encoding a protein of about 100 amino acids and a single transcript. So once I click on sequence, this is what it's gonna pop up, and exons in this case are highlighted in red. So how do I include variants in this sequence? What I'm going to do is I'm going to click on the configure this page link at the bottom. This is going to open up a new inset. And the first thing I want to do is show variants, select a yes. And I'm going to show you how you can have links for each particular variant, so you can go and find out more. But you can have the links hidden. I'm also going to show the line numbering, because I like to see the reference for my sequence. And finally, don't forget to check the check mark, click on the check mark, or anywhere outside of this square to close the window and see how the display has been modified. Now I get to see many of these variants for this gene, and they are color-coded according to the legend up here. If I mouse over an individual variant, I can get a little more information. In this case, the alleles, the name, and the position that they are in a exon. If you see on the left, I'm still on the gene page. I'm on the sequence page, and I want to go next to explore a little bit more about the genetic variation. I'm going to select the variant table, and this is going to bring up a table of all over 900 entries for single nucleotide polymorphisms. I have the option of show or hide the columns by clicking on this feature and selecting or unselecting the options. And I can also filter my list, my long list of 900 by consequence. I can select one of these categories, which is using the sequence ontology terms that I introduced you in the beginning, or I could simply use these box for filtering, and I'm going to use the word stop because I want to find anything that has introduced or lost a stop codon. And I can see that there are two variants for this gene, and the location is, they map apart about 16 base pairs, if I'm not mistaken. No, 14, and which is the same distance. The coordinates are not exactly the same because this is very likely because this publication was using the assembly for TER 9, which I would need to double check, but we are currently on TER 10 on the Ensembl Genome Browser. So we did identify those variants, and now we can just explore whether there are structural variants for these genes in this region. So I go back to my left menu, and I see that there is a link for structural variants. I can tell you already that there are none. So what I'm going to do next, I'm going to choose the variant image, which is what you're seeing in the middle. And I can see that there are long stretches, grayed, sometimes black, that are several nucleotides long in length, and these are actual structural variants. They might not have been included for the gene because they map right at the margins. So if I click on those, I get more information from these boxes, and they are telling me both of these examples are deletions, and there is the allele, which is several nucleotides long. Now, remember I had some tabs here. I also have a transcript tab for this gene, for the only transcript that there is. And what I'm going to do next, I'm going to again look at the sequence options. I have exons, cDNA, and protein. I'm going to choose exons, and when I do that, I immediately get a sequence, the sequence of exons highlighted in big small caps, big capitals, capital letters, while introns are in small letters. And again, the variants are shown as color-coded according to the legend here. Just remind you, this was done using, again, the configure this page, setting the options there, and I can download this, all of these sequences. When I do that, I can get, the next thing, I So that was for exons, but now what I'm going to do is I'm going to choose cDNA, and when I do that, this is the display has changed a little bit, and is including the amino acid sequence corresponding to that to the sequence, to the DNA sequence on top. So I choose as one, and I configure second, and I can, if I click on this on a particular variant, I can see the more information about it. And if I go to explore this variant, it's a link for that particular SNP. But before I go to that to the SNP page, I'm going to show you a little one more thing that you can do with the whole sequence, and this is I'm gonna go to the location region, which you can do that here from here, or you can click on that on the tab up here. So once you go there, you see that it's highlighted, and I'm gonna select the configure this page option again, and now the options are over here. I can select the different variation sets, which in the for Arabidopsis there are about 21. Yeah, I think so. Yeah, 21. So by clicking on any of these sets, I can select how I wanted to display: normal, collapse, or expanded with or without names. And one very interesting tool is how to visualize variation in an alignment, and when you have two sequences aligned to each other. So first you go to alignments, text on the left tab, and then you're gonna select a species that you want to align your gene of interest against. In this case, I chose the closely related Arabidopsis lyrata. And third, don't forget, we configure this page to bring in the variation to our image. This is, in this case, I chose to show the variants with the links. I chose to line numbering, and I want to show conserve regions, so which you will see in these as blue squares. So I can see where in what parts of my sequence there is more conservation, and whether the variants that I'm interested in fall in those. Okay. So remember, I promised that we would go into this link for exploring a particular variant. So I'm back to that to my transcript page, and I'm gonna click on explore this variant, and that link is gonna take me to a new tab. You'll see now we have a new view. So the 5 views for the browser are species, location, gene, transcript, and variation. And if you do some jobs like BLAST or some other tools, you're going to get a 6th view, which is going to list your jobs processed. And you can see that when I explore this variant, this link lights up, because that's the option I'm selecting. And the data that's available for this particular variant is going to be colored here. If you see them grayed out, then the data is not available for this particular variant. In this case, there are some other, so at this point, only Arabidopsis has phenotype data, but other species do not. So the next thing is I'm gonna take you quickly to each one of those icons. So the first one, genomic context, right here, is the second in in position on the left navigation bar, and this simply shows you the SNP of interest that you are coming from, all other SNPs around it. In this case, it's a 5 kilobase region, color coded for these are because these fall under in exonic area, and others are upstream or downstream of the gene, the color. You can also remember that same symbol that we use for configure the page. We can play with to configure the image and add or take the data that is available for that particular region. The next option that we have, data that's available for this gene, is the genomic, the genes and regulation. So when I click on that tab, I see that there are two entries showing that there is a functional effect predicted for this gene, the one we know as stop gain, and also it appears to be upstream on another gene. To define a region upstream or downstream of the gene, we use a criteria of 5 kb at this point. Now, if you want more information, you can click on Show, and that's going to pop up more data underneath here, and you can hide it at any time. Okay. So the next option that we're going to see is the flanked sequence. And we have, when we do this, we're going to see the sequence with the color-coded variants around the gene, the SNP of interest. Next, we have genotype frequencies. When we click, we see a table with allele frequencies on one column and genotype frequencies in another. The next thing we have is sample genotypes. We have a table, in this case, showing that individual populations. And if you click, like in this case, just to for demonstration purposes, I chose one that has only 2 entries, and something is going to more details about this particular population is going to appear below, and these are the 2 accessions that exist for that population. The next is going to be phenotype data. And when I do this, I see that my the SNP is also it has been associated. I'm not sure why is this an apparent duplication. The only difference I can see is that here it's been associated with genes, with a number of genes. You can view it in karyotype. I don't recommend that you do this for this particular example because the number is so large that it wouldn't show up well. But if you have a variant with a few associations, then it is very useful to see where else this phenotype is mapped to. Now I just want to show you a couple of slides that are a summary for that Ensembl prepared as quick reference for the variation data on our browsers. And here you can look them on your own. I'm going to place these slides on our outreach website as well as our video tutorial. The last 2 tools that I'm going to quickly mention for exploring variation data in Gramene is the MART. When you do BioMART, it's a very powerful tool to make customized dumps or extract data given certain parameters. Filter, for example, give me all the transcription factors that have stop gain alleles. How would you identify all transcription factors? Well, you could give a list of particular InterPro domains that are associated with transcription factors, or you can use a gene ontology term for transcription activation. Transcription activity or transcription binding or so on. So you could use some of these criteria and do your best. You can also filter the type of variant you want, like stop gain in this case, and attributes helps you indicate what you want, what type of information you want in your results. In this case, gene stable ID, consequence to the transcript, variation ID. You can select a second set. In this case, I choose genes because I want to pair up the variants with genes. And I'm adding now, tell me those genes with their InterPros. And I have those added to my list. So I went very quickly, but we have we have other webinars that are fully marked on how to use the mark tool. The other tool that I just want to mention is the variant effect predictor. The way to get to this tool is by clicking on the Gramene homepage on the navigation bar, which is on the left, tools, or a link that is on the top of the Genome Browser pages, tools, and this is gonna bring you this table where you can click on variant effect predictor, and you also have the biomark here on top and on the tool page. You can also have your data, your own custom data, run through the variant effect predictor tool, and you can see an example of the formatting that you need, which is VCF. You can click, for example, here VCF, and you get 3 entries showing, the ID, the position, the alleles, so on. And this is and it will predict you once you do that. It will predict you here what's the consequence that is you can expect for your particular variants. So the FTP side, we also have VCF and variation data downloadable in various formats. This is from the front page. You can see those links, and it will take you to a page where you can access either the Gramene FTP or the Plants Ensemble. In the Plants Ensemble FTP server, you have many more text formatted files. For variation, you have GVF and VCF formats. I believe there is a third one, I don't recall exactly now. And just as a legacy, I just want to mention that Gramene was known for the existence of QTL data as well as markers, and we are working very hard to bring those back in. At the moment, this is not actively maintained; it's only kept as it was, but we are working towards bringing those back, mapped to the current assemblies. Anyway, if you still want to look at the legacy data, you can go to the archive by going through to either of these links. Here the navigation left bar from the front menu or at the bottom of that same page. Some of the tutorials that are for if you want to go through what I just showed you and static screenshots, you can see a movie made by the Ensembl team, how to browse SNPs and CMVs. You can also look if you're only interested in structural variation, there is another movie, and there is a documentation for accessing this data with the variation API, as well as we have a webinar ourselves that you can find at the Gramene Database YouTube channel. And this you can get to from the Gramene homepage. We have a link right on top. This is just a sample of what we have. How to reach us, we have, you have email, we have a contact form, you can go to the channel, you can subscribe to our announcement mailing list. There is also a link from the Gramene homepage. You can join us on Facebook, like us, or you can follow us on Twitter. I feel the need to acknowledge the number of individuals very talented that have made this possible in the group of Doreen Ware at Cold Spring Harbor and Dr. and Paul Kersey at the EVI. And two of our external collaborators, Leah Wang, working on the Cybers project, also under Doreen's supervision, and Gary Sanders from EVA, who have worked very closely with us. And the last thing, I also mentioned that I had prepared a very detailed table with data for each particular, each of the 12 species that we have data for, the number of variants you can add up, and you should get over 200 million SNPs. Here, the source and the reference for those studies that where the data came from. And at the end of my slides, you'll have a couple of exercises that you can do on your own and see if you what I just showed you was useful. Okay, so that's all for now, and I'll be happy to take any questions.